Welcome back, guys. Now in this video, let's discuss a topic from the general physiology, that is cellular junctions. Okay. See what exactly are these junctions means. In between the cells. Okay, here look here guys, here in between the cells there is a junction. Okay, which is made up of some proteins. First of all, what is the importance of these junctions? These junctions are bringing the two cells together. Okay, these junctions are bringing two cell membranes together. Okay, so your cells are not simply floating in the air, right? They are not separated with each other. All our skin cells are our body cells, our tissues, our cells are attached with one another, tightly cemented, okay, with each other. So they are your cellular junctions. Let's see what are the different types of cellular junctions. The first cellular junction here I am talking about is a tight junction. Okay, look at the image. Here is a cell 1, here is a cell 2. In between them there is a tight junction present. See, these tight junctions doesn't allow the transfer of any substances from one cell to the other cell. These are very tight in nature. These tight junctions are made up of proteins called as a 𝐾𝐿, Occludens. Okay, 𝐾𝐿, Occludens. Okay, 𝐾𝐿, Occludens. See, within the cell, they are not attached with any cytoskeletal filaments. See, they are not attached. Okay, 𝐾𝐿, Occludens, they are just like a cement attaching two cells together. Okay. Now, in between cells, there are also adherent junctions present. Adherent junctions. These adherent junctions are also bringing the cells together, no doubt. These adherent junctions inside the cell, look, inside the cell, they are attached with a cytoskeletal filament called as actin. Yes, actin already we have discussed. Actin is a microfilament. It's a cytoskeletal filament present in each and every cell. Okay. So, what are adherent junctions? Adherent junctions are the cellular junctions which are made up of 𝐒𝐫𝐤𝐴DN. Okay. So, 𝐒𝐫𝐤𝐴DN. You might have heard 𝐒𝐫𝐤𝐴DN mutations is seen with the gastric cancers. Okay. So, right, adherent junctions are made up of which protein? 𝐒𝐫𝐤𝐴DN. 𝐒𝐫𝐤𝐴DN. So, inside the cell, these junctions are attached with actin microfilaments. Actin microfilaments are cytoskeletal. Desmosomes. These desmosomes are especially present between the keratinocytes. Can you tell me what exactly are keratinocytes, guys? Keratinocytes are the skin cells. Okay. So, your skin cells are attached with one another. Okay. One skin cell, keratinocyte, is attached with the other keratinocyte with the help of which proteins called as desmosomes. So, these desmosomes are made up of proteins. Which proteins? Desmoglin, desmocolin. Okay, desmoglin, desmocolin, desmoplakin, plakoglobin. There are different types of proteins are there, but important are the desmoglin, desmocolin. So let me show you the desmosomes. Look at here, guys. Here is a desmosome, desmosomal protein. These desmosomal proteins, see, within the cell, they are also attached with the cytoskeletal filaments. Which cytoskeletal filaments? Intermediate filaments. Okay, you know, inside your cells you are having different types of cytoskeleton: microtubules, microfilaments, intermediate filament. Actin is an example of microfilament. See, these desmosomes are attached with intermediate filaments inside the cell, inside the cells. Okay, next let's discuss about the gap junctions. So, in between the cells, you are also having these gap junctions. Look at here in the diagram. So, here there is a gap junction which is made up of certain proteins called as connexons. Actually, these connexon proteins, actually there are 6 connexon proteins, okay, going to form this gap junction. Now, importance of this gap junction is it allows the transport of substances from one cell to the other cell. So, which cellular junction is helping in the transport between the cells? It's a gap junction. Gap junctions are made up of connexins. Actually, connexins are the subunit. Connexins forms the connexon. Okay, done. Now, in exams, they will ask the diameter. What is the diameter of this gap junction? What is that gap through which the cells are going to transport the substances? The gap is somewhere around 0.8 to 1.4 nanometer. Very, very small gap. Usually, ions are transported from one cell to other cell. These gap junctions are present in your cardiac myocytes. Gap junctions are present in your neurons. Gap junctions are present in your smooth muscles. Okay. So, gap junctions allows the transport of substances, especially ions, from one cell to other cell. Next, gap junctions are made up of, just now I have told you, connexon. Okay, connexon. Connexons are made up of 6 connexins. Connexins are the subunits. Next, hemidesmosomes. What exactly are these desmosomes means? Desmosomes. Guys, you know what are desmosomes mean? Look here. These are your desmosomes which will attach one cell with the other cell. But you know, our cells are lying on the basement membranes. Our cells are not simply floating, right? They are lying on a basement membrane. Let's say here is a basal lamina or basement membrane. So there are certain adhesion molecules which will attach the cell with the basement membrane. So such adhesion molecules are called as hemidesmosomes. So again important, hemidesmosomes does not attach one cell with the other cell. The hemidesmosomes are the proteins which will attach one cell with the basement membrane. Just like hemidesmosomes, also there are molecules called as focal adhesion molecules. So hemidesmosomes and focal adhesion molecules, they will attach the cell with the basement membrane and the basal lamina. Okay. So this is also completed. Hemidesmosomes, they are also made up of proteins, guys. These are also made up of proteins. Which proteins? Let me write here. Hemidesmosomes are made up of. They are made up of BPAG1, BPAG2. These are the hemidesmosomal proteins. Now you might think how this is important for us. How this is important for us? I will tell you. Before going to there, look here in this cell. These are hemidesmosomal proteins. It is hemidesmosomal protein. It is also attached to some filaments within the cell. It is also attached to one cytoskeletal filament within the cell. What is this cytoskeletal filament? This is your intermediate filament. So yes, desmosomes are attached with intermediate filaments. Also, your hemidesmosomes attached with intermediate filaments. So both the desmosomes and hemidesmosomes, they are attached with intermediate filament within the cells. Desmosomes attach one cell with the other cell. Hemidesmosomes attach the cell with the basal lamina. Okay. Now, look, if this connexon is a mutated, means connexon mutation. I have told you connexons are the ones which forms your gap junctions. So there are diseases in which the gap junctions are mutated. So what are those diseases? Gap junction mutation can lead to diseases. The most important disease is the cataract and the Charcot-Marie tooth disease. These are the two important things which I want you to know. There are other things also like erythrokeratoderma variabilis, idiopathic atrial fibrillation, X-linked Charcot-Marie tooth disease, cataracts and Clouston syndrome. So in all these diseases, connexon proteins are damaged. Okay. In the same way, I have told you, it's the desmosomes. It's the desmosomes which are present between the keratinocytes. Okay, desmosomes are present between the keratinocytes. So desmosomes are attaching one keratinocyte with the other keratinocyte. Now you tell me, if these desmosomes are damaged means, the keratinocytes will separate from each other. Keratinocytes are separating from each other. So there is a gap that is forming. So in your skin, the desmosomes are damaged means the keratinocytes will go away from each other. Okay, at microscopic level, the keratinocytes are separated. So gap is going to form and that gap is going to be filled with fluid, leading to the formation of cyst or leading to the formation of bulla. Okay, so desmosomes, if they are damaged, or if I should say there is any damage to the desmosomes, the keratinocytes are separated, leading to the fluid filled sacs within your skin called as a bulla. Okay, so Now let's look at that integration part. Derma integration, important points. There are diseases where autoimmune diseases. There are autoimmune diseases where autoantibodies are produced against a protein called as a desmoglin. I have told you desmoglin is a desmosomal protein. So antibodies against desmoglin 3 going to produce a bullous disorder called as pemphigus vulgaris. So if you search pemphigus vulgaris images, say there will be bulla to the patient. Okay, it's a bullous disorder. In the same way, there is an autoimmune disease where two antibodies are produced against a protein called as desmoglin 1. Okay, so it is also going to produce a bullous disorder called as pemphigus foliaceus. So tell me guys, whenever you see the word pemphigus vulgaris and pemphigus foliaceus, both are autoimmune disorders where the autoantibodies are attacking the desmosomal proteins. In pemphigus vulgaris, it is a desmoglin 3. In pemphigus foliaceus, it is desmoglin 1. That is affected. Next, so there is one more condition where autoantibodies, IgG, autoantibodies are produced against the hemidesmosomal protein C. Now tell me, when the hemidesmosomal proteins are damaged means, now the cells will move away from the basement membrane. Now the cells are just lifting up. The cells are moving away from the basement membrane. So again there is a gap which will be filled with fluid. Okay. So that is going to cause bullous pemphigoid. Which disorder? Bullous pemphigoid and linear IgA disease. So in these two disorders, bullous pemphigoid and linear IgA disease, there is autoantibody production against the hemidesmosomal proteins. In pemphigus vulgaris and pemphigus foliaceus, these are autoimmune conditions where autoantibodies are produced against the desmosomal proteins. So ultimately at the end of the day, all of them are going to cause the bulla. Okay, they are going to call the fluid-filled sacs. Okay, fluid-filled lesions called as a bulla. Okay, so with this, the important topic for your exam, cellular junctions, is completed. At the end of the day, I am telling you there are different types of cellular junctions present between different types of cells. Most important thing is the gap junction for the exam. Gap junctions are made up of connexins. 6 connexins form one connexon. The diameter is 0.8 to 1.4 nanometers. Mutation, this connexon mutation is going to cause diseases like X-linked 𝐒𝐻𝐫𝐢𝐚 disease, cataract, Clouston syndrome, idiopathic fibrillation, etc. Erythrokeratoderma variabilis, etc. Okay guys, hope the video is helpful. Thank you.